Hi, I'm Suzuki from Nagoya University, Japan. Today I would like to introduce a case study of the application of HARK to conduct a bird song analysis. This is an overview of this talk. First, I would like to introduce why we focus on bird songs, and then talk about HARK part, and also talk about the previous work using this and some problems and how to solve this problem. And then we talk about the one recent studies on the playback experiments and 2D localization of songbirds in California. And finally, I will conclude my talk. So why bird songs? Bird or avian vocalization is classified into two types of sounds. One is a song, like this. This is a long vocalization by males of songbirds to advertise their territory or attract females in a breeding season. And the other one is call, like small faint sound. This call is a shorter and simpler vocalization, which tends to be produced by both sexes and in particular contexts. And basically, it has a particular function. So bird songs can tell us a suite of useful information about the environment, or for example, about the quality of the environment. So the bird songs observation is used for environmental monitoring. And also bird songs have rich and complex variety of structures. So recently, bird songs are used for the benchmark problems for classification tasks. But the bird songs also enable us bird individuals to interact in complex ways. So the bird songs and population of songbirds can be recognized as a complex system. This is our focus, and this is what we want to understand. And so there is an emergent dynamics in acoustic communication among songbirds, and this is composed of multiple individuals interacting with each other using acoustic signals that have various complexity in structures. Temporal sound space partitioning of songs among neighbors is a typical example. So the recording you are listening now is a recording from our experimental field in Nagoya University. In this recording, there are three different species: spotted Narcissus flycatcher and blue and white flycatcher. If we analyze the song timing of these three species, less time was vacant or occupied by two or more species, and more time was monopolized than expected by chance. So they tend to separate the song timing in order to avoid the overlapping with each other, in order to advertise their songs more efficiently. So they are interacting with each other and realize the more efficient use of the shared acoustic space. But in order to this kind of analysis, the detailed annotation and analysis of the recording is required. It is quite costly. On the other hand, spatiotemporal information is itself an important ecological property of acoustic events in natural systems because it can strongly affect their semantics, functions, or roles. Ecoacoustics is a newly emerged discipline that aims at tackling ecological research questions through the lens of sound analysis. There are two roles of sound in this field. One is a subject to understand its properties and functions themselves in the environment. And also, it is a tool for the indirect measurement of biodiversity or habitat quality for the assessment of the environment. The fine-scaled spatio-temporal information can contribute to this field in both aspects. So from this aspect, from this background, and also for the purpose of understanding the complex interactions among individuals in a real field, it is important to obtain the fine-scale spatial temporal information. But actually, the sound source localization techniques using microphone array is recognized as a promising approach for long time for this purpose, but still that is not widely used by field researchers because of the limited availability of both software and hardware. So we are developing a portable system for bird song localization in order to obtain fine-scale directional data of bird songs in a field. This is composed of the laptop PC with an open source software for robot audition HARC and commercially available microphone array, for example, Tamago, which is introduced in this tutorial. And also, it has interface for recording, localization, separation, annotation, and exportation of results for ornithologists, for the researchers of birds. And this is a snapshot of the interface. So you can see some setting parameters for the localization process. And if once you press the button, you can get this kind of result. So this is the snapshot of the localization result. This is the time axis, and this is the azimuth. So that this is the direction of arrival. So that this is the music spectrum, and this is the frequency. So that this is a spectrogram. As you can see that so you can see the some boxes around here. Each box corresponds to the localized sound by using the HARC. So you can see at 0 degree there are several localized sounds, and this corresponds to the songs of this Japanese white-eye. And also you can see some other sound at this direction. So you can, this is the songs of the brown-eared bulbul here. And not just visualizing the result, this application has an interactive annotation tool for the localized sounds. So you can adjust the result in order to correct the result. And you can also delete unnecessary localized sounds and also edit the mislocalized sound or something. And by using this system, we have been conducting some example trials. This is a playback experiment on Japanese bush warbler. We simulated the situation in which conspecific, the same species individual included in a territory of an individual of Japanese bush warbler in an experimental forest in Nagoya University. This is a map of the field, and around here there is one Japanese bush warbler, and this one. And we placed around here microphone array and also loudspeaker here. And we replayed the two types of the songs, the same species song of this species. One is this is a type H song. This is the meaning of the advertising his territory, and another one is this type L song, low frequency song. This has a meaning of the territorial defense against an intruder. So, so we replayed from this loudspeaker these two types of recorded sound, and observed the directional movement pattern of this real individual using a HARC using a microphone array, and also classified the song type of this individual. And this is a basic finding. This is a directional distribution of the song of the target individual from the microphone array. This is a 360, so directional distribution of the histogram of the localized sound. So as you see, without playback condition, you can see that this individual tended to sing here and this direction and this direction with two types of songs. On the other hand, when we replay the song of the same species, you can see that this individual tends to sing only to type L songs, and this individual tends to move actively around the loudspeaker. This is a typical behavior of the aggressive response. Actually, when we conduct the similar type of the experiment with different conditions, if we assume the more aggressive condition, we could observe the more active response. That is more large directional change and more frequent vocalization of type L sounds. So we can qualitatively obtain this kind of the we can quantitatively obtain this kind of response. And this is another example. The benefit of using this approach is that we can localize the vocalization of birds at night. So this is an example of localizing the nocturnal vocalization of scaly thrush in our experimental forest. In this case, we use the 60-channel microphone array called DACHO. So this allows us not only estimate the azimuth, but also estimate elevation. So this center corresponds to the sound coming from top, and this outside area corresponds to the sound coming from the horizontal direction. And anyway, so what we can see is that at night, this species flew over the microphone array, calling repeatedly. This kind of behavior cannot be observed visually at all. So this is quite interesting. So we have introduced two example trials, but it looks that the Harvard system works very finely and they easily obtain the information of bird songs. But actually, we need much effort to obtain that kind of result because many and various sounds were localized in the field recording in general. So the recording contains other bird species vocalization and sounds of insects and water flow and so on. So actually, this is an example of the all localized sound in the first experiment on the Japanese Schwabra. So each white box corresponds to the localized sound. So there are many. But actually, what we need, so for our research purpose, is just these colored sounds. So we need to extract the sound sources of interest from them. This is very costly to do this manually. So in order to solve that, we have added one function to the application that is an annotation on the space of the spectrogram. So we use the dimension reduction algorithm called T3, which is a very popular dimension reduction algorithm, to create the map of the spectrogram of the localized sound. So by using this, we mapped this spectrogram of localized sound onto the 2D-dimensional space. So each dot corresponds to the one localized and separated sound. So as you see, we can get some clusters of the sounds, and this application allows us to replay each sound by clicking each dot. So we can easily listen to these clusters around here, and we can recognize that this is the type H song or Japanese warbler, and this is type L song, and this is the other species song, and this is a water flow, and these are the cluster of the other faint unknown sounds. So we can easily annotate that these are the songs of this target individual, and this is quite important for our purpose to annotate the recording in field. So by using this, we first conducted experiment on the 2D localization and classification of songs of spotted towhee. To see how our system can quantitatively grasp the spatial dynamics of bird vocalization in field, we place the system with two microphone arrays in the territory of two individuals of spotted towhee in May 2018 in California, and we replayed the songs and calls from a loudspeaker. This is a map of the experimental field, map of the field, and this is a territory of target individual A. These are the my two microphone array. This is a loudspeaker, and this is for the target individual B. And this is a song of this spotted towhee. Where is a call replayed from the loudspeaker? When is this is a song of this individual. So anyway, we conducted the recording and we could observe the 10 song types and one call. Actually, there are many types of the songs in this species, and that song is individual specific. And we replayed the specific type of song and the call of the individual A for every 2 minutes, and for it, and we conducted the 60 minutes recording for each setting. So song of individual A against A, or call of the individual A against B or something. So in order to obtain the spatial information, we have to combine the information from the two microphone arrays into one. This is quite difficult, not easy. And so we classified bird vocalization in the recording of two microphone arrays using the interactive annotation tool based on the t-SNE. So this is a space map of the localized sounds from one microphone array, and this is a map from the another microphone array. And as you see, and there are some clusters, and colored clusters correspond to the vocalization of individuals and the vocalization types. So you can easily see that this and these are the same type. We have to classify it manually, but quite easily identify the cluster. And then we extracted a pair of sources in the same classes from two arrays that have any overlap in the localized time frame, and the DOA information of the pair of source was used for the triangulation, like this. Please look at our paper for detail. This is an example of the spatial distribution of vocalization. For the this is one microphone, and this is another one, and this is a loudspeaker. And green dot corresponds to the localized location of the loudspeaker, sounds from loudspeaker. So that's very close. So the localization process was very successful. And you can also see some clusters of songs. This is all the experimental result of the spatial distribution of vocalization for each condition. And anyway, what we could obtain is that target individuals tended to be more actively responsive to playback songs than calls. For example, this is a playback condition, and this is a song playback against individual A and call playback against individual A. As you see, this is a frequency of the responded to sound from the target individual. So actually, clearly, so you can see that when we play the sound, when we play the songs, the individual tended to respond more frequently, and this color corresponds to the song type, so they tended to respond with various song types. And also, this individual tended to be closer when we play songs, and also this individual tended to change the direction largely when we replace the songs. So all the tendency, that is, this individual responded with various song types at closer and more different locations, and reflect the their behavior is more aggressive. So we could quantitatively extract the spatial dynamics of bird vocalization successfully. So in conclusion, the spatial temporal information is an important factor for a deeper understanding of complex acoustic interactions among songbirds. Hark and Hackbird can provide quantitative and fine-scaled spatio-temporal data of bird vocalizations that cannot be obtained by human observation. As a future work, we have been conducting the analysis of acoustic interaction in the population of multiple birds. Actually, we are using this kind of mesh tent, and in this tent, we released several individuals of the zebra finch, and they can freely fly around, fly in the tent, and we have trying to localize the sound vocalization of them, and trying to estimate who is singing and who, where, and who is singing, and what type of the vocalization. I'd like to thank all the collaborators for this project here, and I'd like to finish my talk. Thank you very much for kind listening.